everyone, and welcome to the Balanced Bodies Blueprint. I am your host, Vinny Russo.
And I am your co-host, Dr. Erin Stansfield.
And we're shifting gears from all the conventional fitness narrative you hear on most fitness podcasts, as our main emphasis lies in preventative healthcare, adopting a holistic approach to nutrition, and challenging the traditional views on various fitness topics.
Our mission with this podcast is to provide you with the information you need to achieve optimal health.
And on today's podcast, we have a very special guest. He's known as the Cancer Daddy, aka Cancer Zaddy. His name is Dr. Joe Zundell. So, Dr. Joe, thank you so much for joining our podcast. I know this means a lot, not only to our viewers, but definitely a lot to Dr. Erin, as you guys share common ground with the cancer. So,
Yeah.
thank you so much for coming on.
Yeah, and thanks again for having me. I really appreciate you both, reaching out and inviting me onto your podcast. It's been good to get to know you both, even in a brief time that we've met each other. So seriously, it's an honor to be here on your podcast and take a bit of your weekend time up.
Oh, well, you're giving your weekend too, so.
Yeah, we all are super busy, have very little free time to do this stuff. So it's nice when people try and accommodate my schedule as I try and accommodate theirs too. It's always a two-way street.
Of course, of course. So why don't you do this? Why don't you, for those of our listeners that don't know who you are, why don't you give a introduction of who you are, what you do, so they have a better understanding of what we're going to be diving into in today's podcast.
Yeah, absolutely. So my name is Dr. Joe Zundell, but for most people that know me, I'm not really into the titles or anything like that. So please, anyone who's here, just call me Joe. I am by training a biologist through my undergraduate training. I did that in St. Leo University in Florida here in the United States, just outside of Tampa, for those unfamiliar with the area, very small town. So I got my bachelor's of science in biology, and I have some personal underpinnings into, my interest into cancer biology, but lack of a story short story here, just moving on. I've been interested in cancer biology for a while due to my own personal circumstances. We can get into that later. That led me into pursuing a degree in cancer biology, which later I moved from St. Leo, Florida, or St. Leo University in Florida, and ended up moving to Philadelphia as a technician after working briefly at Moffitt, the cancer center in Tampa, moving to Philadelphia, and then eventually about a year and a half into moving to Philadelphia, got into the University of the Sciences and the Wistar Institute in their cancer biology program, and so I do cancer biology. That's what I got my PhD in, and it's a pretty vague description for something that encapsulates all of the biological and mechanistic chaotic things that can happen within cancers. That's the foundations of my education, such that I can better understand the disease and from a preventative and a treatment perspective. So hopefully that keeps it simple for people.
Yeah,
That's
it
great.
definitely does. Definitely very layman there. And you could have definitely went a little bit more into stuff, but I could see the humbleness coming out of you right now.
Yeah.
But so you touched on it for a second, and you said we could get into it a little bit later, but why not? Let's just get into it now.
To it.
What drove you into cancer research?
Yeah. So it's not something that I talk about lightly. So basically, when I was 14 or 15, my mom got diagnosed with esophageal cancer. And that's pretty tough to deal with as a pretty young person. Um, and she when you watch that disease, particularly esophageal cancer, is kind of bad because it's seemingly hard to detect because the symptoms associated with esophageal cancer are kind of common. And I come from an Italian background, so it's like, we eat a lot of pasta sauces, and my mom had acid reflux, and these are kind of common things that kind of mask the symptoms for diseases like esophageal cancer. And so, when things kept progressing and giving my mom worse symptoms, she, was seeing doctors, and they were like, "Oh, just take these acid reflux pills and heart or blood pressure medications and heartburn medications and stuff," and things were not resolving. She saw a different doctor, eventually got an endoscopy, camera down the throat to check for any signs of other issues, and they found pretty late stage esophageal cancer. And so my mom eventually passed away when I was 17 from that cancer. And when you watch the progression of that disease, um, it's life-changing. And I've dealt with it pretty heavily over the course of my education. And it's been really depressing. But I mostly felt a lot of anger when I was going through it just because I didn't understand it.
Mhm.
A lot of times when people go through a cancer diagnosis, even just as an observer, as I was, you just question why, you know? And thankfully, I grew up in an environment to recognize that things in life are not simplistic. Um, and I knew enough not to just stop at the WebMDs and the Googles and to actually pursue an education if I really wanted to understand the meat of the disease. So watching my mom die essentially from that cancer stimulated my curiosity and wantonness to cope with that loss. So the only way that I could deal with it without, believe it or not, and this might be a little hard to hear, but without like killing myself, um, was to pursue an education so I could physically understand and feel at peace. And I will say that the only time that I felt peace from the amount of anger and hopelessness that I felt in watching that situation was the second I stepped out of my door, out of the door for my thesis defense. Where I literally, it was just like a decade of pain. And it's like, it's just hard to talk about. It was the only time I felt peace.
What a beautiful
Yeah.
journey, right? Like I know
Sorry to get
it's so
emotional. It's,
No, it's perfectly fine, and
it's
you have every right
yeah,
to feel
yeah.
that emotion come out.
And
yeah,
we started this podcast because before we had this like pre-conversation, and I am here to be so grateful, right, to people like yourself. Um, what a wonderful journey to actually, and what a purposeful life to have, to see something so tragic that affected you so deeply, and now to respond with an intellectual journey that is difficult,
Yep.
right? And that may have not even answered the underlying questions of that why, but to
I mean,
help
I can,
others fight that battle, right?
yeah,
So we started this because I wanted to thank you for the work that you do, because it may have not saved your mom's life, but it saved people like me. I had cancer as well. We talked about this briefly when I was 16, and people like yourself saved my life. And so I am forever grateful for people like yourself that may not even get the recognition that they deserve. But you guys saved my life, and Western medicine is can be a blessing. Let's put it that way.
yeah.
It can save people's lives, and we may not always know why, right? My cancer, we don't know why it happened. I had a acute promyelocytic leukemia. I still don't know why, and my mom still struggles with the why, right? Why did it happen? What could I have done to prevent it? Are there, you know, earlier tests that we could do? And that's kind of what brings us to the topic today is cancer testing. But I just want to display my gratitude for your beautiful journey that has been difficult and dramatic. And honestly, I think you should write a book. I think
I, I've toyed with
I
the
would definitely
idea.
read it.
The amount
It's
of, the amount of things that I've been through, that's, uh, it's life, right? So like we all go through these hoops and these hurdles. Um, Not that that was an easy thing that I've been through, but there have been a lot of times where in my life, like if I go like as an adult now, having looked back on things that I went through when I was younger,
yeah.
I just think, how the fuck did I do that?
Yeah.
Yeah. I can't
And there's
imagine going through that at that age too. That's such a critical age.
and that's I
Now
mean, that's the tip of
having
the iceberg for me. There's a
yeah.
whole lot more than that too, and it's just yeah. So maybe I should write a book.
You definitely should. And maybe we should have you on again for actually a personal note story.
I'd be happy to talk more about my past and how it enables me to um, do what I do. Because I think anybody who's ever worked with me will tell you that I'm probably the hardest on myself than anyone ever is around me.
Yeah.
Yeah.
And there's a pretty big reason for that. I have often stupidly high expectations, uh, severe perfectionism, um, and it can be debilitating. And thankfully, at least now I'm around people that help bring me down to baseline and say, "Hey, Joe, stop kicking your own ass. Biology's
Yeah.
hard enough.
Yeah.
Yes, yes,
Just do the work,
yes.
and we'll figure it out."
Well, and know that people like myself are eternally grateful for the work that you do. And like alluded to this before, but it can often be a thankless job. And we
Yeah.
appreciate your presence on social media as well to communicate that to everyone. That's also a tough job. And knowing that you're out there, that you're working in the trenches of cancer research is commendable and will always be. So again, very much grateful for the work you do. But
Yeah.
let's get into
Well,
the
Yeah.
cancer
before
thing.
before we got into that, I just have to say, like with your journey. So if you asked Dr. Aaron, I'm a very positively spin everything you can, right? I'm always doing that. It just helps me
Yeah.
cope with a lot of things too.
Absolutely. I
And with
make a lot of jokes about things I probably shouldn't just to get through the times, you know?
well, yeah, I mean that it could be someone's way of just getting through stuff. But the way
Yeah.
I'm looking at this is like what you went through is not something that should be wished upon anybody, right? But what you did turned you into who you are today, and what you're going to do with your career, which might save millions of people, right? And that's the positive spin that I want to put on this journey. It's like you went through a very, very hardship you wouldn't wish on anybody, and not saying that it was like a sacrificial lamb, but that happened to, and it caused you to become who you are today, which is only going to further progress into what you will become in the future. And I believe that you're gonna uncover some shit, man, that's gonna save millions of lives, man. So that's the positive spin I got to put on your journey.
Yeah, no, I appreciate that. And the thing is like ego plays a pretty big part here, but I will say that there's nothing special. I mean, there are special things about me and the way that I do things for sure, but nothing I do matters if I can't work well with others. And this was something I've learned relatively recently. Like I said, I'm a often debilitating perfectionist, so I get very good at doing things myself, and it leads to trust issues of other people. But the thing is, like, I can't do everything myself. I don't have enough time to do everything myself. And even if I know I'm going to do something perfectly, I have to enable other people to learn and also learn how to do something up to my standards as well. And so I might be able to do something that impacts the human race in terms of, my research and stuff like that too, but it cannot be done if I cannot work well with others. And so it's not just me. I think a lot of people need to understand that this is a group effort, and this is the humility thing that,
Yeah.
maybe is coming out a little bit too, but
Yeah.
like this is what enables me to be effective in what I do,
Yeah. I think
especially as I progress in my career.
Yeah.
yeah. I think it's rare for a scientist to admit that, and thank you for even saying that. We can relate on in more ways than one.
Yeah.
Let's put it
Yeah.
that way.
Yeah. And, it's why I left academia.
Yeah, I can share a similar sentiment with
Yeah.
with
Most people can.
perfectionism and that working alone is my preferred method, right?
Yeah.
It's difficult to actually work with others and then also to communicate the way that you communicate. And again, it is a challenge, and we're grateful for you doing it every day.
Yeah.
Yeah.
So with that
Well,
being said, let's
yeah.
dive into the science now.
Yeah,
Um,
buddy.
Yeah. So with cancer and cancer research, there
Hmm.
is something that comes up pretty often now, which is
Hmm.
called the Gallery test, right? Gallery
Yep.
testing. So I feel like this is going to be a big component of what we talk about today. So why don't we start off with you just explaining what gallery testing actually is?
Sure. Yeah. So the Galleri test, I think that's how you say it, or gallery test or whatever. It's
You would
this
know better than
tech.
I. So
I Actually, Grail is the company here. It's actually here in North Carolina where I live.
Oh, nice.
Actually, before I was moving here, I applied to the company just because I think it's great technology,
Yeah.
Yes.
and the research behind it would also suggest that it's pretty great technology,
Yeah.
Yes.
and should even be FDA approved within this year, next year,
It's great.
Yeah.
maybe.
Yeah.
But we'll see. It's basically this. It's a liquid biopsy. So what the whole purpose of this test is to detect cancer in earlier stages, because as I'm sure you guys know, and even some of your audience, the earlier you detect cancer, the better the prognosis, right? So the more likely we're able to treat the cancer and increase cure rate, so to speak. So basically, the way that this test works is when you draw the blood, you purify circulating DNA. And so when cancer cells get a little fucked up, so to speak, they often release their components, and oftentimes this includes DNA, into the proximity of around that tumor, and sometimes it gets into the blood. And depending on the abundance of that DNA, you can get this kind of like fingerprint, so to speak. And
Mhm.
so the way that the Galleri test works is that they use epigenetic signatures to produce this fingerprint based on signatures derived from normal cells of the same tissue lineage. So every tissue in your body will have a specific epigenetic fingerprint or signature. And when a cell decides or adapts to become malignant or benign, even in some cases for a cancer, its epigenetic fingerprint changes. And so this test is designed to detect those fingerprint changes between a cancer cell and a normal cell that enables us to better to detect the cancer. And it uses AI training modules to do this as well.
Wow.
Does that make sense?
Yeah.
Yeah. Yeah. That's
That's awesome.
incredible.
Yeah.
Yeah,
Can
it
you
is pretty
just,
neat.
yeah, I think it's amazing and I'm excited for this, which is why we're having this conversation.
Yeah.
Anything to detect cancer earlier, I think would be beneficial.
Yeah.
Can you tell us a little bit how it might be different from the traditional single cancer screens like a PSA or a mammogram or something like that?
Yeah, so the primary difference is it uses sequencing technology. Um, like a PSA test is based on, total abundance of a protein, but the turnover rate of proteins is somewhat more unpredictable and less stable than detection of DNA within a person's blood.
Yeah.
And so, as I'd mentioned with the Galleri test, it is an AI model trained system, so it needs a lot of information, which is why it's gotten better over time. So it's just like any education. The more information you provide to a model or a classroom, the more capable the students are going to be to understand what you're trying to teach. So this is the same as their AI model. So it's able to work more effectively in a single test because of the materials that are examined from it through its sequencing methods, but also because it's a massive learning model that is able to detect these different signatures that it's been trained on between different cancers and normal tissues. So it's not just looking at PSA, it's not just looking at HER2 expression, or it's not just looking for KRAS mutational signatures. It's looking for a bunch of different things at once.
Yeah. And
And
just
that's
so
why it's accurate in many cases and it's inaccurate in some other cases.
Yeah, the way I look at it, and correct me if I'm wrong, it's like we're trying to see at the DNA level versus some of these other tests are, miles down the road, if you will, with the actual expression of the gene or the protein that's happening. So, if we can actually catch it at the DNA level, then we might catch it sooner. That being said,
Is that accurate?
That is pretty accurate. I will say it's at the DNA level, but from a slightly different perspective. And so remember, going back to this, I mentioned that it's like an epigenetic signature. So the way that they're looking at this isn't directly through mutations that could occur in a cancer. Of course, the assay is better or more accurate in detecting cancers if they're later stages, because obviously there's more cells, there's greater likelihood of detecting the stuff. But it primarily works by detecting what are called methylation patterns. And so
Not Gary
when
Brecka methylation that he talks about, right?
I mean, it is what he talks about, but not in the way that he talks about it. In other words, I'm not here to preach bullshit that doesn't make any sense,
Right.
like Gary Brecka.
Yes.
Yeah,
Go
that guy
that
can
guy.
fuck right off. Um, but anyway, so like when we talk about epigenetics, it's not just methylation patterns, right? So like when we think about methylation when it comes to epigenetics, typically when a specific sequence of DNA has these methyl groups added on to histones, it's associated with genetic repression. So a gene is turned off. So there's histones where DNA is wrapped around, and the methylation status of those histones and those genes enables the DNA to be wrapped around those histones such that they're not open and capable of being expressed into those functional proteins. And so what enables a gene to be expressed is often the inverse of methylation, which is typically considered like acetylation. So those methyl groups are removed, genes can be acetylated, and then that decreases the interaction of the DNA from the histone. So think of this like a ball of yarn. Like we're unraveling the yarn from like a ball of yarn when we acetylate the histone, and that enables the genes to be accessed such that they're going to be expressed into proteins that do functional things within a cell to complete a job.
Okay.
And so this is a very dynamic process, epigenetics. So
Yeah.
like, and it's also super quick.
Yes.
So that's why when we think about methylation status of genes, it's not just methylation. There's monomethylation, there's dimethylation, there's trimethylation. Does it occur at a promoter? And now I'm getting nerdy and kind of like
Yeah, I was
jargony,
just
right?
gonna say now you're delving into the complexity of like, so people throw out these terms like methylation of DNA,
Yeah.
but please explain to us how complex that may look like.
Yeah, and so these methylation or acetylation patterns can occur at promoters. They can occur at enhancer regions, and depending on where they occur, genes aren't always in an on or an off position. They can be intermediate. So the way that I tell people this, it's like a dimmer switch. You can have intermediate levels, either low or high or somewhere in between of a gene being expressed, and these are always changing depending on the environment. So it's not so simple to say, oh, a methylation signature is indicating this specific cancer. It has to be validated beyond that to ensure that you have an accurate representation of a diagnosis, in this case with the Galleri test. So maybe you're going to ask this at some point, I'm not entirely sure, but this is why typically the Galleri test alone isn't sufficient to diagnose a disease, despite it having pretty good accuracy in a lot of the cases. It's almost always, and it should be paired with some other diagnostic to confirm what the assay is saying. It's like if I was a scientist, I would never do one specific type of an experiment and say, okay, let's put this drug in cancer patients now because I did this one very specific experiment and now I'm 100% confident that it's going to work in humans. We have to do things from various different angles to validate that we're actually observing the right thing.
I think what you just said is so extremely important, right? Because we get very excited about what this test might be and oh my goodness, like if you can get a blood test and or so to speak a liquid biopsy
Hmm.
and not have to undergo other testing. I think that's what people think. I won't have to undergo other testing. I would just caution and say, hey, this isn't the greatest screening test. And we can get into like specificity, sensitivity, that sort of thing.
Hmm.
But it needs to be coupled with the whole clinical picture. And oftentimes that's going to be a clinical history, a clinical exam, and even further testing. Sometimes imaging is going to be involved in that whole thing. So it's
Yeah.
not just a one stop, although we would love that, right? We
Yeah,
would love one
absolutely.
test to like test everything. But
Hmm.
if you really are concerned and you are using this as a screening test, I think it's too early to just say this is a great screening test. The numbers aren't there for the sensitivity and specificity.
Yeah, and I had a
Yeah.
quick question too, because you
Hmm.
did say that the test is AI power or AI driven.
Hmm.
Do you find that it hallucinates a lot? Because that can run into some complications.
Um, so it always depends on how well the model is trained.
Mhm.
And it can, right? So it can produce false negatives or false positives. It depends on the stage of the cancer that's detected, right? So
Mhm.
like that's why it's important to get other experiments done or other imaging techniques or modalities performed so we can have confidence in the results to confirm that it is a positive or a negative.
Yeah.
Because again, everything that we do educationally with AI is about how well it's trained, right? So if it's trained on a relatively limited data set, because let's say there's a lot of cancers that aren't as common as breast cancer or lung cancer. We already know the epidemiology behind all of these things. The cancer isn't as abundant, your learning model is affected. So it's going to affect the accuracy of that. So yes, so the way that we get around this is by using other assays or modalities, whether it's imaging or a bunch of other things to
Yeah.
validate our observations.
Yeah. All right.
Yeah, it's
Thank
a good question.
It's a great question. He's done some AI work, and
Yeah.
so he's seen
Seeing it
AI
hallucinate a little bit.
hallucination.
Yeah. Yeah. So
I
that's
use
crazy.
it.
Creating an app
Yeah.
and seeing it like hallucinate, and you're like, come on, why? Like, why are you doing? I'm almost there. And it's like you got to go back and rework some things. So
Yeah, it's
Yeah, no, I do this regularly. I use this I do a lot of histology in my work, so I look at a lot of human patient samples and we have these things called tumor microarrays and it's basically you get these like little punches of tumors and you put them on a glass slide and it's from like hundreds of different patients. And I used to do this all by hand and by eye and staining for specific protein expressions and then fucking counting
oh geez.
it was horrible. So I did this in grad school and it's like just screwed up my eyes, but like
I can imagine.
we had a shitty microscope too.
Uh,
And I sat there with like a counter, one of those
Oh man.
counters and
sounds awful. It sounds like a grad student kind of project, though.
yeah, and we told the PI that was like, dude, we have and this wasn't even like we're in modern times. He could have
Yeah.
bought software, but like in academia, sometimes the funding is limited. You have
Right.
to be very careful how you
Yeah.
spend it. But and I get it. But the thing is, like, now I'm using this software called Visiopharm, and it's awesome, and it utilizes AI learning models to identify like what of these cores of cancer patient samples are actual cancers.
Mhm.
And then when you stain for a specific protein of interest or a marker of interest, as an example, how much of it is there. And so we have to train it on normal tissue a lot of the time. And sometimes I'll train it and it'll regress.
Oh
And
no.
then I'll have
Yeah.
to continue training it on more samples of normal tissue to make sure that it increases the accuracy that it's detecting the thing that
Yep.
I'm trying to understand. And it can be very easily biased because I'm not a pathologist. Like I'm not a trained pathologist. It's not my career. So I have to like work with actual human pathologists to say, am I making the right conclusions from
Yeah.
these
Yeah.
observations that the AI is making? So it's just fact checking from all these different angles. And a lot of people will use AI or like chat GPT and they'll be so confident in the materials that are spit out. And it's like you still have to
Gotta look
validate
them
it.
up. Yeah.
Yeah,
Yeah.
yes, 100%. Yeah,
Yeah, because we
Yeah.
were we were actually doing something like PMIDs, and it was giving out PMIDs
oh,
that
those,
didn't
yeah.
make any sense. It's like I don't even think that research
Yeah,
exists, and then
but
you'd go to it,
when
and it's
you
like
like cross-check it, you're like,
it's,
oh,
this isn't
that was a
this isn't
different
the article.
article.
Yeah,
Yeah.
yeah.
Yeah, it
You
does
can
it all the
evidence a little
time.
bit better for that. But
Yeah.
anyway, sorry, we digress about AI, which is
What
one
you
of
point
my
out here
other.
too is like other AI are better at some things than others. And
Yes, I was,
again, it's
yeah,
because it's how they're trained.
Yep.
exactly,
Yeah.
exactly. Anyway, back to I'm gonna mess up I'm gonna call it the gallery test, and so I'm gonna just refer to it as the Grail test. But it has a high specificity,
Mhm.
99.5%. I think it's been cited in some studies.
Yeah.
Um, how does it compare, would you say, to other blood-based tests and minimizing patient anxiety? And I know I alluded to this in saying, we need to talk about the specificity and sensitivity here. So can you kind of give us an overview and maybe you know how you feel about the testing and where it's at?
And just to throw this in there, if you know, great. If you don't, just pass it. Which cancer actually has the highest? Which cancers have the highest sensitivity? If you know that, that would be great to know.
Yeah. Yeah. No, I do, based on the literature at least. And this is one of the things that makes the gallery test, the Glary test, um, well sought after and trusted is because it's currently the one that has the most research behind it. Grail has put a ton of money into ensuring that They conduct clinical trials. I think it's the Pathfinder series of trials that they're conducting. Um, so they've put a lot of time and money into ensuring that their tech is doing what they say it does. You don't want to end up like Elizabeth Holmes situation where you're producing. You guys familiar with Elizabeth
Yes,
Holmes?
with the one drop, and
Yes.
now
Yeah.
she's still in prison, right?
I believe so.
I would hope so. I hope
Yes.
she's never allowed out. That's horrible, right? But yeah, so like Grail doesn't want to end up in a situation where they're making promises they can't keep, right? And so they're trying to collect as much data as possible behind their assay. And so how it compares to a lot of other companies is that other companies are also using similar cell-free DNA. But the way that it varies between companies is they might be looking at different metabolic signatures or epigenetic signatures. Because remember, I said epigenetics is more than just methylation patterns. So a lot of different companies are doing different things. Hell, there was even actually, I sent it to Vinny this morning. I don't know if he shared it with you, but there
No,
was
we
a
did.
paper
We looked at
published.
it. Yeah.
There
Yeah,
was a,
and please get into that because it's an interesting concept, right? Not only
well,
looking at cell-free DNA.
yeah, and this gets into the question that Vinny asked a moment ago, which was, like, why is this assay better at some cancers than others, or is it better at detecting some cancers than others? And so this paper that was published the other day, and I didn't read all of it, so you'll have to excuse my ignorance in this, but like,
We didn't
um,
either. We looked at
it's
it.
a Nature
But it's
Cancer
cool.
paper, and
Yeah.
it's a lot to digest, right? So like these
Yes.
bigger journal papers, and I've published in Nature Cancer once. It's kind of challenging to do, but
Congratulations, that's a huge accomplishment.
that's not a flex. There's a lot of
I
politics
think it's a
behind
flex
publishing.
because maybe it's the nerd in me. I'm like, oh, that's amazing.
Yeah, there is a lot of politics behind publishing, but we can get into that later. Anyways, um, basically, this new paper uses a combined approach to better understand and provide accuracy and precision to the cell-free DNA approach that wasn't there before with the Galleri test. Um, so essentially, the Galleri test isn't perfect, right? It has high accuracy, high precision in terms of detecting cancers, but there's some cancers that it doesn't do a good job of.
Right.
And again, I mentioned earlier that depends on the staging of the disease
Mhm.
and often the type. So it does a really good job, at least accurately, with detecting things like pancreatic cancer and liver cancer. And these are great because we don't have great treatment options for these things, especially here in the United States, in the clinic.
Yeah.
So it's detecting some of these more lethal cancers early. Colorectal cancer is on the rise. It does a great job at detecting that.
Mhm.
But it doesn't do such a good job at detecting skin cancers kind of globally. It doesn't do a great job at detecting prostate cancer. So this is one of those situations where maybe instead of using a Galleri test or doing the Galleri test, you'd have to pair it with a PSA
Mhm.
Right.
antigen test.
Yeah.
Um, but this new assay builds further on to what Galleri is doing or Grail is doing and provides greater depth so that it enables better accuracy into detecting some of those cancers that it wasn't so good at before. So like melanoma is improved, prostate cancer detection was improved, those sorts of things. So that's how this kind of works now.
Well, and what I gleaned from that Nature paper was that they're essentially building like this huge library of
Hmm.
signatures
Yeah.
from the different cancer types, and it's not necessarily. I guess it could help with a clinical test down the line, but it was more like here is the library of what these cancer fingerprints might look like, what these unique fingerprints might look like, and
Yeah.
very academic in nature, if you will, but also very
Yeah.
exciting, right? Like
Yeah.
that these different cancers might have different fingerprints and unique, DNA fragmentation uh,
Yeah.
that
And
that
this
that
is
will be
these
evident.
experiments that were in that paper are something I did quite commonly in my grad school training because I was trained in an epigenetics based cancer metabolism research lab because one of the proteins that I studied was a pretty strong epigenetic regulator, a pretty potent epigenetic regulator, at least in normal cells, but gets a little bit fucked up in cancer cells, obviously, such that it's been named as a tumor suppressor. And this is the the protein ARID1A. So one of the assays that we often do in my experiments, at least for epigenetics-based research, is called ChIP-seq. And so it kind of builds on, you know, getting better signatures by utilizing these purified sequencing methods, looking at different types of epigenetic marks, again, not just methylation patterns, to better inform a larger data set, as you pointed out. So that's what's enabling greater accuracy in these signatures is we're just gathering more information and it enables better training.
Yeah. Well, and since we are talking about your research, can you tell us about your current research? And I know that you can probably go into radiopharmaceuticals, that sort of thing, but tell us about your current research because we know that this Braille test is going to be something here in the next couple of years. I think it's
Sure.
going to be exciting. We need to pair it with more testing, but tell us about your current research and what you're working on.
So you want to talk about, so I have phases of my life where I've done differing types of research, and I try and stay versatile in my approach because it's obviously more impactful for what I'm trying to do. But so do you want to talk about like what I did slightly pre-grad school, grad school, or do you want to talk about what I'm doing now with radiopharmaceuticals?
Let's start with your postgraduate stuff, and you can even talk get into what the challenges were there. I know that you alluded to that before our podcast, and I think it's important for people in non-science backgrounds to
Hmm.
understand like maybe some of the politics that go on in academic situations and not only is the science challenging and actually getting the work done, but also there's politics involved with that, especially in academia.
Yeah.
And then maybe
Then clinical.
platform
Yeah.
into what you're doing now because radiopharmaceuticals. So I did nuclear medicine training.
Yeah.
We used to stage cancers, so we did the PET-CT stuff. And so we were always looking for what therapeutics did the patient get because it would drastically change our PET-CTs, right?
Yeah.
So anyway, so yeah, so if you can start with your post grad and how that platformed you into what you're doing now and what you are doing now.
And be honest about your feelings.
Yeah, yeah, yeah. Please.
Express.
Yeah.
I
So
asked my videos going through that.
you don't have to ask me to be honest about my feelings. It's often gotten me in trouble, but I refuse to be anyone now that I'm not.
Yeah.
So if that's hard to hear for some people, I'm sorry, but I'm not sorry.
Too bad.
I will
Yeah,
be respectful, but I'm going to be honest about the things that I experienced. And sometimes it's a hard fucking pill to swallow.
Yeah, but that's authenticity, and that's what we
we
respect.
love that.
Yeah. And it's also why I don't have a shit ton of followers on social media, but it is what it is.
Yeah,
Those people can follow Gary Brecka.
yeah, exactly.
Um,
I
yeah,
think there's something to be said for intellectual integrity, and you have that. So
I appreciate that. It's
we will shout that from the mountaintops.
Yep.
I appreciate that. It's something that you would think is easy to hang on to, but apparently not so
Oh, it
much.
is
Not so
not.
much. And listen, in my realm with coaches, you see them fall off like flies all the time, just trying to find new business, drum up new business, and going off
Yeah,
on these tangents that don't even exist and are not science-based or evidence-based. And it's sad to see it happen because
yeah,
you've got some really good coaches that are just they need to make money. So they go to the dark side, for lack of a better term. And it
yeah.
sucks, but it is what it is.
Yeah, not to go off tangent here, but believe it or not, when I started my Instagram page, before I even knew you guys many years ago, when I was a couple years into grad school, my social media, my Instagram page was a fitness page.
Oh, really?
Oh, that's
Yeah,
cool.
so the reason that I kind of started gaining followers because eventually I got Lane Norton's attention. You familiar with Lane Norton?
Yes.
Oh, yes.
Yeah, and now we're good friends. Like we talk regularly. But I was initially reviewing supplements
Oh, okay.
because a lot of these
Interesting.
I've gotten like cease and desists from Muscle Tech and
Yeah, yeah, yeah. That's
like when you
great.
display that there's no fucking data about all this
Expose
shit that
them.
they're saying.
That's awesome. We
Expose them.
actually recently got asked by somebody to review a supplement, and I told Vinny, I'm like, why don't you just tell them their supplement shit, like
Yeah.
from the
Yeah,
very beginning? Because there's
yeah,
there's a there's a few studies out there that are actually valid to say the supplement is good, right?
yeah.
There's maybe like two supplements that ever that
That you would approve of.
yeah that I'm like, oh, there's actually enough data to say, yeah, this one might actually make a difference.
So
But
creatine and what else?
I'd say creatine is probably the main, and protein.
Protein powder, creatine.
Me too. Glad we're in
Protein and
agreement.
creatine, and that's about it.
Yeah. So you don't
That's
want people injecting glutathione or
No, 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 none of that. None of that.
unless you want to
it's
give me an aneurysm.
Yeah.
yeah,
Oh,
it's
unless you're dealing with heavy metal toxicity.
yeah.
Right,
Those are
right.
the ones,
Oh
right?
God.
Yeah.
I just that's a whole nother tangent that we could go into.
I
But
mean,
yeah,
Aaron
I
and
Very,
I might
yeah.
have more like actual heavy metal toxicity just because of the work that we're in, but most
Yeah.
people do not.
Yeah.
You know, what did you
yeah.
deal with? Copper 64, fluorine 18 or something? Or
Yeah, yeah, and well, it's funny. So actually, my specialty is occupational medicine besides
okay.
preventative medicine. So this is my preventative medicine side, but I actually deal with actual heavy metal toxicity every day.
Okay.
So my patient population works with like cadmium,
Oh,
hydrofluoric acid, like major no-nos, right? And
so a
so
lot of veterans, I'm sure.
yes,
Yep.
yes,
Yeah.
exactly. So this is the aeronautical, aerospace kind of industry. So they're working on airplanes and engineers and stuff like
Yeah.
that. So they're actually coming across some bad toxic things. So I think it's funny when I see influencers on social
Oh
media and
gosh.
they're like, "Are you being poisoned?"
By lead.
And it's just like, are you kidding me right now? First off, the dose makes the poison, and guess what? Toxins are
It's
everywhere.
funny because she's going to be on the Docs Who Live podcast talking
Oh,
about that
nice.
specifically. So
Yeah.
yeah.
So,
Have fun
yeah.
with
So,
that.
yeah, yeah. Anyway, sorry.
Wait, so supple, you were
We
going into it. So
digress.
yeah, so you had a fitness page, and then you were getting all these cease and desist by all these supplement companies because you're calling them out and they're bullshit.
Yeah.
Yeah, essentially. And then I obviously I don't have any money for fucking lawsuits, right? So I was like, I gotta stop this because
Yeah.
apparently I'm rubbing people the wrong way. And so I was in my education, I was like, you know what? I'm just gonna like teach people cancer biology because you go at least at that time and even kind of still you go on social media and you're like, no one's like actually teaching stuff. You go to like the National Cancer Institute and it's just like a blurb and it's like sign up for my newsletter to learn more, but nobody's gonna fucking sign up to your newsletter.
Yeah.
Like how
Yeah.
many
Yeah.
people do you think wake up in the morning and be like, God, I wonder what the National Cancer Institute's gonna send me today? Nobody ever. I even commented on a post, I think it was like the Pancreatic Cancer Network. They did the same thing that the NCI does. And I said, you guys, just describe it here. I'm not going to read your caption and then
Right.
feel so inclined and motivated when I've got this vague thing that you've just presented to
Right.
Yeah.
go sign up to your newsletter.
Yeah.
Yeah.
Like it's not action worthy. If you want
Right.
action, just fucking explain it.
Just
Yes.
go, you
Yeah.
give it
And so
out. You
that's
give it
what I
out for free,
started doing.
you'll get the
Yes.
traction.
Yeah.
If you give it out for free, you will get the traction.
I was just gonna
Yeah.
say, I so much appreciate what you do. It's like you bridge the gap between, the hard sciences of what's going on in cancer in the cancer world, right?
Yeah.
To everyone that can access social media, and I can appreciate that because as a cancer patient, I always wonder where cancer research is, or as a former cancer patient, I should say, I always wonder where it's at. But my medical specialty, even for me, I don't know what the latest and greatest is in oncology. I'm not looking at that constantly. And so for
Mhm.
somebody like you to bridge that gap and be like, oh, listen, there's, this new study, or this is what's going on.
The
Um,
article you
then
just sent this morning.
then I can look it up and say, oh, I'm going to read about that, because this is a new and interesting topic, or a new discovery, for example. I saw something about CAR T therapy, for example.
Mhm.
So my type of leukemia, I remember after going through, my year in and out of the hospital and almost dying, and being in ICU, and it's horrible. And
Right.
all your family that's watching, that's horrible for them as well. I remember thinking, when I got better from that and going into medical school at the time, they didn't have a better treatment option. And I remember people asking me like, "Hey, if you got that again, would you undergo treatment?" And I said, "Absolutely not. I know what the probability is of a cure, and I don't have a bone marrow match. They wouldn't find one in time, and I'd probably die from something like DIC. And so I knew what my options were
Are
if
you
it
talking about for CAR T?
yeah, well, So then CAR T came out,
Oh, I
and
see. You're talking about like
yeah,
stem cell transplant.
yeah, yeah. Well, and or a bone marrow transplant, right?
Yeah.
At the time, that's all they had.
Hmm.
But then CAR T therapy came out, and it was people like yourself that, were putting stuff on social media, and I was like, oh, what is CAR T therapy? Again, my specialty is not oncology, and so I went and looked at it, and I was like, wow, that would save my life today if I happen to,
Relapse.
have a reoccurrence of APL, which could potentially happen. The probability is never zero.
Yeah.
Um, I, that therapy would likely save my life, and it's not it's different from chemotherapy, right? It's a different
Oh yeah.
treatment.
No, it's a completely different modality. Yeah, I guess we've deviated from your initial question here a little bit, but yeah, no, like the if your cancer were to relapse today, you'd have so many more options
Right?
than you would have had when you first were diagnosed. And it's,
Yeah.
even breaking away from CAR T, basically you take your own T cells out of your body. It's a type of immune cell that has anti-cancer action, so to speak. You take it out, you basically genetically modify that T cell to make something on its surface that recognizes something specific to the cancer that you want that T cell to target. So you're essentially creating like a little guided immune cell missile.
Yes.
And
Yes.
then they're manipulated, which is not very easy, and it's not always successful. They're manipulated outside of the body to express that chimeric antigen receptor, which is CAR. And then they're injected back into your body to go seek and destroy the Metallica song, right? So like,
Cool, man.
um,
That's so
yeah,
cool.
It's I think it's amazing, right? Because my traditional route of chemotherapy now has a different option. And that option has a higher probability and likelihood of actually being successful than repeat chemotherapy round.
yeah.
And so,
But outside of this, and sorry to interrupt you, there's so
no,
much more than CAR T. There's other types of immunotherapies. There's immune checkpoint blockade now, which targets like different immunosuppressive proteins on cancer cells. There's even more targeted. So you told me this previously before we even started the episode, but you had a chromosomal translocation
translocation.
or
Yep.
fusion, and I don't know if this is the case, but like we have drugs now that do better at targeting those fusion proteins that are made from uh, different sort of chromosomal abnormalities that can happen in a lot of blood-based cancers. So for like CML or AML, oftentimes a lot of people have heard of the Philadelphia
Yep.
chromosome, right? So it's
923.
also a translational or chromosomal fusion translocation event between two different proteins. One of them is BCR and the other one's ABL, and it makes this weird hybrid protein when they fuse. And this is where, Gleevec, a drug, came to market and
Yes.
targeted that fusion protein that's not made in normal cells. So it was like the first targeted and selective therapy to something that was totally cancer and had very low likelihood of targeting normal cells because of that targeting to that fusion protein. So like nowadays we even have small molecule chemotherapeutics that are better at targeting those fusion proteins as we've learned more about various diseases of cancers over time.
Yeah. Well, and anyway, you communicate that to people, and I love that. I love that you are out there in the general population and saying, hey, guess what? This is an important topic. I mean, cancer is in the top five killers in the United States, right? Of mortality risk. And so um, everybody knows somebody that has been affected by cancer or even died from
Yeah.
cancer.
Hmm.
And so I think the work that you're doing is so important, especially on social media.
Yeah.
So,
Well,
um,
it's a new way to get information out,
yeah,
and you're doing that, and we
Yeah.
really appreciate that. We
yeah,
do.
yeah,
Well, and it's kind of going back to one of the questions that I was supposed to answer here. I think that was maybe
we've
like
Post
we've
grad
20
research.
minutes
we've
ago. But the thing is,
digressed.
like,
Yeah.
the whole reason I wanted to, I mean, well, maybe not the whole reason, but like in my career, I wanted to become a professor. I wanted to have my own lab. And so I went through this academic route to be able to teach people because I do think that one of my superpowers, so to speak, is teaching ability.
Mhm.
And it's not without effort, right? And so I put a lot of time and effort into just like, and this is why it takes me forever to make posts, to just try and like have people look at things. I review my own wording and I say, if I didn't know what I know, does this make sense?
Yeah.
And I'll send it to my friends. I've worked with a bunch of people who are not scientists that I've made friends with over the past, couple years that I've been on social media. And I've been like, hey, you're not a scientist by trade. Does this make sense to you? And if yes or no, if somewhere in between, how can I make it even better? And it takes a
Right.
shit ton of time, but it's peer review. And that means everything to me. And
Yep.
so I quickly
That's
learned as I progressed in academia that it's not about, especially in bigger institutions, and I'll talk about this. It's not about how good of science you do all the time. It's about who you associate with more often and the pursuit of clout that enables your success.
mhm.
Um, I'm very bullheaded in the way that I do things, if you couldn't tell, and I have a very strong moral and ethical compass. And I'm not saying that everybody in academia is bad, but in my association in the environments that I've been in general, the science that people do is often not trustworthy because it's in the pursuit, or not fully trustworthy because it's upon the pursuit of clout more than it is academic integrity. So we can publish all these fancy mechanisms and papers in Nature Cancer, not that that one's bad, but we can all publish these fancy things in papers, but at the end of the day, it means jack shit if it's not reproducible. And
Mhm.
I've been in so many situations where I always live by the principle trust but verify.
Mhm.
It has nothing to do with like me having personal vendettas against people. It's you did this experiment. I trust that you did it correctly, but I also have to do it, one, to learn, but also to verify that I'm not putting myself into a trap where I'm ending into this publication landscape where I'm potentially going to publish something that's not entirely true. So it has to be reproducible. And
Yeah.
so this led me into my postdoc when I graduated, which was a very short and depressing 8 months. And essentially, I realized UPenn is a pretty horrible institution. Um,
Well, and we
and
can we can say similar things, right? Of we call them
yeah,
in medicine, we call them malignant programs. I don't know if you guys use the same terminology, but
pretty accurate.
yeah,
Yeah.
yeah.
It
And
goes hand in hand with the
I
cancer.
had a similar
Exactly. I was just going to
Yeah.
say that. It's a
Goes
good
hand in
pun
hand.
for the episode.
yeah, I had similar experience at Harvard when I was doing some of my nuclear medicine training. And
Yeah.
so um I think I think knowing these challenges for somebody who's not in a science field, to know how difficult it is to actually put in that work and what kind of challenges are even um, encountered as far as like the politics of academia, and not only that, but what you're even alluding to as far as like what actually gets published, right?
Yeah.
And what doesn't get published. And so these are all very big challenges. And so when people, when that's filtered down to the person that's like, um, given a PMID number, for example, and they're like, hey, this study showed this, and some influencer is saying that, I think it's very useful for people to understand that they need to be skeptical, right? You need to
Hmm.
take it with a grain of salt. And then the amount of training that actually takes to interpret that PMID and to know where that came from and what kind of politics might have even influenced that paper getting
Yeah.
into that journal, right?
Yeah.
Is so
I've been on
important.
so many papers where somebody's added at the last minute, like this big professor, and it's like they
They
didn't
didn't do
contribute to
anything.
this paper. You're
Yeah.
having their name on this paper
Yep.
because they've got a big name, and oh, now you think your fucking review process is going to be easier, and it actually is. And that's depressing to
That's
watch
sad.
over and over
Yes.
again.
That's sad.
Yeah. Yeah.
Yeah. Wow.
And so
It doesn't
people,
affect the science. Like the science can still be good in that paper, but that actually takes away the quality from it.
Yep.
yes. Yeah. Well, and it takes away sometimes like what actually is published, right? Because
Yeah.
sometimes if you don't have that name on there, it may not make that journal. And like you said, the science might be solid. And so I think that people need to understand. I like what you said. You have to verify. You have to, that's part of being science. And if you can't do that, then you need to find somebody who does do that, right? Like you need to be getting your information from somebody who is qualified, who is filtering through that information, and who has the intellectual um, you know, intellectual capacity, but also the integrity that's going to be there. And I think
Yeah.
that's hard to find
Well,
unless you have
it's
the
hard.
training.
It's maybe hard to find, but going from my postdoc experience at UPenn, which again, I do not advise postdoc or grad school at UPenn. Undergrad is good,
Yeah.
but postdoc and grad student for sure. My wife did her grad school at UPenn and similar multiple
Experience.
horrible
Oh,
experiences.
gee.
Wow.
And it took her 7 years to graduate. But the thing is, like, when I started my postdoc, fresh out of grad school, I started doing these experiments to try and, reproduce some of the observations that a previous postdoc had published uh, in Cancer Cell, another
Big
great journal.
journal.
I love
Yeah.
Cell.
Yeah. Cell's a
I
great
think
journal.
Cell does a better job than Nature most of the time in terms of not only explaining science, but also providing better quality and reproducibility. But um, I was doing these experiments in a way to not only reproduce the observations from a previous postdoc, but do it in a more physiological way. And I
Mhm.
was using a relatively simplistic approach, but it still required a lot of critical thinking about, specific assay conditions such that I could observe this thing physiologically because you can overexpress something. You can stress out a cancer cell to observe something, but that doesn't mean that it's real. And so I
Right.
was trying to understand it under more real contexts.
Yeah.
And I was unable to do so through multiple different angles. Remember, I said you have to go through different angles to validate something. I figured maybe I'm not approaching it correctly from this angle. I'll try all these different nutrient deprivation conditions. I'll try doing this in hypoxic chambers without oxygen to see if like there's different metabolic contributions that are contributing to the thing that I was trying to observe, and I didn't see it. And at the end of the day, I kept telling the PI, the person that led the lab, and it just consistent condescension.
Yep.
You're not doing this correctly. All you've done here was a couple Western blots, and you've not really achieved anything. You're not asking good questions. So and so in the lab can do it. Why can't you? And it's like, okay, what do you want me to take away from this? And then eventually, because I wasn't reproducing this postdoc's unreproducible shit that was published, I was forced to sign a resignation letter.
That's
Forced
insane.
to resign. Not willingly,
Yeah.
forced to resign.
It's insane. That's
So,
and it's sad. Honestly, it's sad. It's their loss, man. Like to
and
me,
no ego, I say it is their loss.
yes.
When you have somebody like I worked with someone, I'm not going to name any names because I still respect this person and they've done phenomenal science in their career. But this is somebody who's a National Academy of Sciences member.
Wow.
If you have somebody that's in your lab that's willing to question
Yep.
and do rigorous work to reproduce other people's things, whether you recognize it as rigorous or not, if you're not providing them an environment to do a good job, maybe at some point you have to look in the fucking mirror and think, maybe I'm not providing this person the right resources. And that's the number one thing I learned at UPenn. Nobody gives an absolute fuck about collaboration.
Yeah.
Wow.
Because a lot of scientists will be like, oh, we'll collaborate with all of these groups and then you'll get in the lab. And they'll be like, actually, we're just going to have you sit at your bench in the dark where there's no fucking windows.
Yep.
And you're just going to do everything by yourself. And we'll call you an independent scientist because this postdoc training is supposed to be about you becoming an independent professor. And it's like, I already know how to critically think. I can
Yeah.
do your fucking job. I've
Yeah.
already written grants.
Yeah.
Just make me a professor.
Yeah,
Yeah.
yeah, yeah.
I'll
I
learn that way.
yeah. Yeah.
Anyway,
I
so I resigned, and now I'm in biotech. I have
yeah.
more money. I have more resources. I'm expected to do like truly rigorous scientist or science. And guess what? My work is significantly more likely to actually fucking help cancer patients.
Yeah.
Not this mechanistic clout bullshit that academics seek. It's actually going into the clinic.
Yeah.
Yeah.
It
Well,
it
and
sucks when you when an agenda is put in front of actual science.
well, yeah,
It's
and
it's
I feel
publish
like
or perish. It's the publications that ruin, in my personal opinion, it's the publication pressure that ruins the integrity of science. Because nowadays it's gotten so bad. Like those Nature papers, that Nature Cancer paper, how many figures were there? Back in the day, it used to be like four or
Oh,
five,
yeah, I
six
was just
figures.
gonna
Now
say
there's
you got
like,
lucky if you got one.
yeah, now there's like 8 or 9 figures,
Yep.
and all those 8 or 9 figures are A through J. And then there's another 6 or 7 supplemental figures that nobody ever, no, nobody reads.
Yeah,
Yeah.
yeah.
And so
And
it's overwhelming.
yes. Yeah, just to give you an idea, like I think that when I, did
I'm
residency at
sorry, I
Harvard.
got really passionate there,
No, no,
but
I think it was great. When I did residency at Harvard, like my counterparts had like 20 publications, for example, and I had
bullshit.
I had my measly one, that I worked really hard on. And so there was this publication pressure. Even in medicine, we see this is my point that, and especially at some of these, big name schools,
Mhm.
Harvard, Penn, that sort of thing, that it's like it's like they've lost uh, sight of what the actual goal is to actually help people, and
I think so, yeah.
they're so intertwined in their own ego and how good they are as an institution, and their name. And so it sucks, and then they lose
Yeah.
good people like yourself,
No, they
to
force them out
yeah, they force them out.
in my case.
Yeah.
It
And
all,
then now to private industry.
yeah, it almost reminds me a little bit of what's going on in the education system. So, like, you have this publishing pressure here, so that's your North Star, and you'll do
Mhm.
anything you can, sacrifice quality to get published, right? And it's like with the education, it's like everyone's teaching to a test. So
Yeah.
that's the standard, that's the North Star. You have to do good. These students have to do good on this test, and it's like you lose a lot of aspects of education to get to that score, and it's diminishing the quality that is being provided to students. It's diminishing the science that's being put out to the public.
I have a story here too, specifically related to that. I grew up in Western Massachusetts, and Massachusetts love their standardized tests.
Yeah.
I have never been able to do well on standardized tests. I also realized uh, relatively later in life, when I was, 30, that I have ADHD. But like when you're growing up in a really small
Yeah.
town, it's just kind of taboo. Like mental health just
Yeah.
goes out the window, nobody gives a shit.
Yeah.
Especially as a young boy.
Yeah.
Yeah. Yeah. And it's just like, oh, he's just weird, you
He's
know?
just a boy. He's just wired up. Yeah.
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 Yeah. And so I had trouble paying attention in a lot of classes. I admittedly I'm not good at math. I cannot do math. And you're like, well, how are you a scientist if you can't do math? They go hand in hand. I can only do math if it's applied to science.
Oh, that's cool.
I can do physics. I
Yeah.
can do nuclear. I'm in nuclear medicine right now. I can do all like biochemistry, McKelly's metakinetics. I can understand that stuff because my brain skips all the fucking variables that you learn about in algebra, and it's like, okay, I can apply that logic to science, and in my brain makes sense of that. But I've never been able to do well at math. So growing up, I was put into essentially the special education programs. They thought I was mentally challenged.
Yeah.
And so I took all these classes in math to just try and catch up, and I just couldn't do it. And it's because of this premise of standardized tests. You have to be able to pass this standardized test. And I thought I was crazy for the longest time. And then I just was like, fuck all of this shit. I'm just gonna do things my own way. If
Yeah.
I can't pass the test, I'm just gonna go in the door a different way.
Yeah.
So
Yeah.
like my ACTs to get into or my ACTs to get into, you know, undergrad flopped, did shit. My GREs to get into grad school were fucking abysmal. So I established other ways. The way that I got into grad school was not because of GRE, because nobody would look at that and be like, this person's intelligent.
Yeah.
I got bad scores. I'm not even like I'm embarrassingly not even going to tell you what I got on the math. But basically,
How'd you get
I
in?
thought at that point I was like, okay, I can't pass these tests, so I'm just going to have to show people that I can do
Yeah.
this stuff.
Yeah.
So I got the lab training. I went into the lab and like I can just apply this stuff. I got a publication before I went into grad school as a second
That's
author.
awesome.
Yeah.
That is amazing.
And it was that was the worst working experience I've ever been in. This will also be in my book, I guess. You can read about it
I
in
I was
probably
just gonna
20
say
years.
you definitely need to write a
Yeah.
book. I think your story is so
Oh,
unique, and just so you know, as so I'm on the spectrum, and my child is on
welcome to
the
the club.
spectrum. Yeah, I know, right? And my child is on the spectrum as well, and so it's so interesting because he is on the spectrum, and I'm going to get a little personal here. And he's, he's in IEPs and special ed. If that's basically a fancy politically correct for saying he's in special ed.
Yeah.
And we just found out like his working memory is on the lower end. And it's so funny. I was mentioning this to my mom, who said, "That's crazy because he can tell you like years of every
King,
coin
queen, and
and countries and
where
their
countries are.
capitals are. He knows more countries than I can even point out on a map,
Mhm.
right? And he knows their capitals. He knows like what years certain coins came out, like what monarchies
That's
can be on that
awesome.
coin. But so she's like, how can they say that his working memory isn't great when he can know so much about that one topic?
Yeah.
And so just to your point, standardized tests aren't the be all say all.
They shouldn't
And
be.
not only
No.
that, but look at where you're at now. Like you have PhD. Look at the research and the life changing things. And if you had believed that at a young age and hadn't
I
figured out a different route to go around it, you wouldn't be where you're at. If you would have said, "Oh, well, I'm not good at math, so I'm not even going to
Well,
try."
yeah, you did what teachers are supposed to do. You made it relatable. You made it relatable to what you actually enjoy, right? So you took math and you integrated it with the science that your brain was working with, and it made you understand that math. And that's what
no, no, no.
teachers
So
have to
here's the
do.
thing, like, and with your child as well, it's like I just fucking love life. Like observing. So I started my career as a marine biologist. I love the ocean.
Yeah.
Yeah,
I would still be a marine biologist if I could. I have tattoos of
that's awesome.
jellyfish. I have, this is a
That's so
Yeah.
testament. This is
cool.
the anglerfish that was
Yeah,
discovered in
Nice.
yeah,
2000. This was
yeah.
a science paper. That's why I got this tattoo.
Yeah.
That's awesome.
Like, I love the ocean, but
Yeah,
I love life.
yeah,
And so I can't see myself like physically and mentally doing anything else.
Yeah.
right.
And so I had to do that. And so a test cannot capture that sort of
No,
Exactly.
can't even.
obsession.
Yeah,
It's an obsession.
It's one
yeah.
of the reasons why I left teaching. It wasn't because like, well, the main reason was because the nutrition business just took off and I
Yeah.
didn't need to teach anymore.
Congratulations.
Thank you. But it was mainly because like after COVID, like we weren't able to fail kids and they just kind of gave up on like putting forth effort and all these dynamics started changing and everything was driven towards a test. And I worked at a behavioral school. Every kid had an IEP. So you had to teach these kids individually very differently. And everything was just
What's
getting
an IEP?
it's basically like
Individualized education.
well, that's the name, yeah.
Yeah,
So basically you go through and you say like what they're having issues with and then what your solution to
Oh,
that issue is.
yeah,
So for example, if you have ADHD or
yeesh.
whatever and like maybe
yeah.
you need a little bit longer time to complete a task or something like that.
That becomes really challenging to teach people, I'm sure.
Yeah,
Oh,
it
yeah,
is. But
right.
everything was being so pushed down into this like bottleneck of like standardized tests and have to do good in it. And I'm like, these kids are having trouble read you're just passing them along. They have third grade reading levels in high school. I'm like,
Yeah.
I'm not going to push them to a test. I'm going to teach them the way I want to teach them. And you got to get on different levels with these kids. And it was becoming too standardized for lack of a better term. And I was just like, I'm done. Like it doesn't resonate with me anymore.
Yeah,
Yeah. And so like, I've had so many bad mentors in my career so far.
yeah,
But I've only had a handful or so of good.
Yeah.
yeah,
And both of those different dichotomies of experiences have enabled me to communicate that the way that I do in teaching people. And I think that without the fear of failing, like if people the if people cannot learn how to fail, they won't be able to acquire the skills in teaching that I currently have.
right. Yeah,
It just enables an intellectual adaptation that enables you to better teach people.
Yeah.
And so like the good mentors that I have had or have been so impactful in what I do and also so have the bad because the what not to do
right.
lessons
Yeah.
are often more transformative than the what to do lessons.
Right.
And believe it or not, like even no matter how good of a mentor you have in science, you get more what not to do lessons because the biology fails you more often than the mentor does.
Yeah.
Right. Right.
Or the biology tries to hide from you more often than anything else. So like, yeah, it's just intrinsically challenging. So I think the best mentor to me has just been life.
Yeah.
Maybe that sounds philosophical, but like,
I love it.
Yeah.
Again,
yeah,
that's a quote for the book.
Yeah.
yeah.
Definitely.
I think
So
a
then
lot of
going,
people would agree. I'm
yeah,
sure you
yeah,
guys would as well.
Yes.
100%.
Yes.
Yeah. So then going from your post grad, and let's go now into, because I don't know how much time you have left. Let's go more into what you're currently doing right now with your research,
Yeah,
so everybody could get a catch where
yeah.
you're at now.
So I've wanted to get into the nuclear medicine space
Oh, nice.
probably since as long as I can remember. Like
Resonates with you,
I have,
okay.
Yeah,
Yeah.
yeah.
I've
I know some people.
yeah. Well, I've had this passion since, like I mean, when you grow up reading about Marie Curie and the atom bomb and all these things, it's like quantum physics and quantum mechanics are just it's so cool to be able to observe these essentially unobservable things or
Right.
low
Yeah.
likelihood observable things
Yeah.
on such a small scale and produce conclusive things about how life works from a radioactive decay perspective or even just how light works and all that stuff. It's fantastic. Um, but also to realize that radiation can be causative for cancers, especially things like lung cancers, when you consider like radon exposure as an example, which is one of the primary causes of lung cancer.
Yep.
So get your houses
I
detected
was just
for
gonna say,
radon.
make sure you have a radon
Okay.
Yeah,
I need
detector.
big
that
thing.
soil.
Yeah,
Yeah.
absolutely. It's uranium decay into radon, and radon gases can get trapped in basements. We don't have basements here in North Carolina, but obviously they do in New Jersey and other places. But
Yeah.
you check anyways, independent of that, because you don't know like how close you are to uranium deposits or whatever. It's fine. But yeah, so there's this double-edged sword with radiation, right? So like it can be causative to cancers, but it can also be used to treat and detect cancers. So earlier you mentioned positron emission tomography, which is
Yep.
PET scan, or maybe people have heard of. And essentially you're using radioactive materials and their decays because they're radioactive to be detected by an imager to find cancer cells within the body. And so I've always been fascinated by those imagery techniques, and now I work at this company. It's called Actis Oncology, and basically we make targeted mini protein nuclear warheads. It's the
Okay.
best, simplistic way I can
I
describe
love
it.
how you
So
said that.
we
That's
make
great.
these small proteins that recognize something, hopefully, as much as we try, hopefully specific on any given type of cancer, and we attach basically like a chelator, this thing that traps a radioactive metal to that mini protein that's guided to the cancer. And it delivers that radioactivity right to the cancer as opposed to systemic radiation, which killed Marie Curie. She got cancer.
Yep.
Yes. Yep.
And a lot of people who worked with radioactivity back in the day, even on the Manhattan Project, fucking just died horribly.
Yeah.
Not even just from cancer, just from like organ failure.
Yeah.
Um,
Well,
Also, safety standards have changed significantly since then. You can't just
Yeah.
manhandle uranium with your bare hands or polonium. But my company specifically works with this radionuclide called actinium-225.
oh,
So,
yep.
yeah, good, you're familiar with it. But for the people who are not, believe it or not, there's different types of radioactive decay. And those radioactive decay schemes, as they decay, they can induce different types of damage if we're using it therapeutically. So, actinium degrades or decays by what's called alpha decay. And I'm not going to go into the specific quantum mechanics of this, but there's different types of decay, right? So, like alpha decay is, at least from what we know in the research, more likely to cause double-stranded DNA breaks when it interacts with cancer cells, as an example. There's beta decay. So maybe people are familiar with PSMA 611 or Pluvicto for prostate cancer that's been approved. That's a beta decay molecule, and those are more likely to induce single-stranded DNA breaks. And so the biological difference between double-stranded and single-stranded breaks and the way that cells repair those are different. And so when it comes to cancer killing, we have to think about what type of decay of a radioactive isotope is going to do the greatest amount of killing for that specific type of cancer.
Mhm.
Mhm.
And it's also cool because what we do now is called theranostics. So while I can have this actinium molecule in that chelator of that mini protein, I can switch that actinium out for something that doesn't kill cancer cells. I can switch it out for copper 64. I can switch it out for gallium 68. I can switch it out for indium 111. I can use different imaging. So like for copper 64, I can use PET imaging to basically detect that cancer diagnostically. And then if I want to make that treatment into a therapeutic, I just switch the radionuclide to actinium. So one is used to detect, it's the same molecule. One of the radionuclides detects the cancer. I switch it out, and one of them kills it.
So it's therapeutics and diagnostics all in
Correct.
one.
Exactly.
That is so cool. And what was the term that you used again?
Theranostic.
Theranostic.
Yeah,
So
I
just
didn't
so
coin that, obviously.
I had to look that one up,
Therapy
but
and diagnostics.
yeah,
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 fair enough.
it's a beautiful term,
Yeah.
and again, this is where your expertise comes in. This is something that's new in this space, right? And so I think knowing that those things are even available, for example, clinically speaking, a lot of docs are familiar with radiation, the damage that does to patients' bodies, right? And so
Right.
if we can target that and minimize the damage there that's done, it will decrease the amount of um, you know badness that happens, or the feeling at least of how they feel when they're going through that therapy, because anybody who's been through that kind of therapy knows that it can be fairly daunting.
Yeah.
So I think anything to target that therapy would be great.
I think
Yeah,
it's
and
pretty
that's
good.
why we're trying to do what we do, right? So like I will also say you can't have too much of a good thing, right? So like the more we've understand about quantum medicine and nuclear medicine, there is this aspect of cancer, right? So let's say we use one of my companies developed medicines that I can't explicitly talk about, obviously, because, I'll get in
They're
trouble
proprietary.
for now. We do have some public information that you can go to, our website for my company's page and look at like some of the targets and the things that are in the clinic now. Um, but basically, there's this double-edged sword with radiation, right? Like you can have damaging radiation, but it can also, when in the case of cancer, it can enable the cancer cell to adapt. So you want your radioactive material to kill the cancer cell as quickly as possible without inducing other adaptive characteristics of that cancer cell such that it becomes resistant to the radiation. Because a lot of people think when they think about resistance to drugs, they think about like small molecule chemotherapeutics, like they're just pushing the chemotherapy out or they're just developing different metabolic things to adapt to the treatment. But with radioactive therapy, it's the same sort of thing. So independent of it being like a protein guided thing or no matter how targeted it is, cancer cells will adopt different mechanisms to respond to that radioactive decay and enable ways to continue to survive around it. So we have to think about those things as well when it comes to like dosing, right? The dose makes the poison. We have to think about that when we make these therapies. It makes it a little bit more challenging.
Yeah, interesting. Well, continue that wonderful work.
Yeah, I
Yeah.
thought it's pretty amazing how,
Yeah.
like, because if you're talking about like double stranded, like destroying the double strands of the DNA versus the single strand, I'm like, go double all day. That should kill all the cancer cells, right? No, not
But it's not that
necessarily.
straightforward.
Yes,
Yeah,
exactly.
it's more
Like,
Yeah.
nuanced,
yeah,
just like
yep.
everything in science.
Yeah.
Complexity.
Yeah,
Yes.
yeah.
Yeah.
Yeah. Because of that in that case, it depends on whether or not that specific cancer, if you know, um, like I said, the responses to deal with single-stranded breaks are different than those
Yep.
than dealing with double-stranded. And if that cancer cell has a propensity or an adaptation that enables it to better deal with double-stranded breaks, and you happen to be inducing those, maybe it's not favorable, right? So maybe you'd want to switch out for something that's maybe a beta emitter.
Yep.
Yeah.
And so like you look in the literature now and a lot of nuclear medicine people that are developing these targeted therapies, they're considering the differences between, in this case, do we use a beta emitter? In this case, do we use an alpha emitter?
Yeah.
You know?
That's so interesting. Like, uh, for somebody that got trained in nuclear medicine, and what you're talking about is so much more in depth, where we're just looking at these scans and going, what stage of cancer is this in? So my work sounds almost so elementary compared to what you guys are doing. I think it's incredible work.
Oh, well, it's
So.
actually not because I couldn't do what I do if people like yourself and other nuclear medicine scientists didn't provide me with the foundations to know what we currently know, right? So like that's the thing that I mentioned
It's a
previously about
team. Yeah.
collaboration. It's
Yeah.
teamwork. I
Yeah.
can't do
It
what I
is.
do
Yeah.
without all the amazing work of other people like yourself and even Vinny here. So
Yeah. Yeah. Well,
Throw me in the mix there.
I like it. I like
I learn a lot from you too. Like so this is the same like I said, I got involved in the fitness space when I started my
Yeah.
page on
Yeah.
Instagram.
Yeah. I
I'm
think
still
that's
obviously interested in them. I've got the biceps to show for it, but
yeah, yeah.
you know,
Well, resistance training will always be a thing. And speaking of that, actually, knowing all this cancer stuff, all this research, all this, you could probably tell us more about cancer than anybody that I would know. What would be your advice to lay population as far as like how do we prevent cancer?
Oh,
yeah,
that's a
it's
loaded
always going to
question.
sound simple. What's that?
That's a loaded question right there.
Well, I know that he has a good answer to this one.
Yeah, I'm sure. I'm sure I have a good answer. No, yeah, I always try and keep this simple, right? So it's like control the controllables, right? All this biohacking shortcut shit, it's not going to work.
Can you say
And
that
so when
again,
I
please? Nice and loud. Say that one more
yeah,
time.
control the controllables. All this biohacking shortcut shit is not going to work.
Thank
So
you.
anybody who's involved with Vinny's fitness programs, get ready to do some fucking work because none of the other shit is actually going to help you as much as Vinny's programs.
Thank you. Thank you for
You're
saying
welcome. And I
that.
don't even work with Vinny, so I have no uh,
You have no
affiliation
skin in
here.
the game with
I have no
helping my
skin in the
business
game,
goals.
right?
Yes.
Um, but yeah, so like the things that, enable good health, they're already riddled with seriously complex metabolic epigenetic underpinnings alone. So, we live in a time where jargon's applied to things to make people like feel smart, and adopting specific behaviors, right? So like I can tell somebody go to sleep at regular, consistent intervals for like 6 to 8 hours every night, and as a human being.
Yes.
And additionally, online you'll hear align your circadian clock with physiological responses that govern metabolic homeostasis
It
associated with light
sounds very
cues,
fancy. Yeah.
uh, you know, enable optimal health outcomes, reduce disease risks, whatever. And basically, both of those things mean the same thing. But
Yep.
people follow the second one and not Dr. Joe Zundell's advice. Go to fucking sleep
Yeah.
at a normal fucking physiological time associated with being a human being.
Yes.
Yes.
No, we have to align our circadian clocks
Yes. Yes.
with light cues and a bunch of research that I actually haven't read. We have to do all that fancy stuff. So like, yeah,
I
I don't know what's going on these days, but essentially the same they mean the same thing, right? So
I
One
think
ugh,
one sounds sexier than the other,
I think the
but
yeah,
basic
it's the foundation
yeah.
is that that's what works. The basics work.
Yeah.
So if you want to call it, if you want to throw it on like all those bells and whistles to make it sound sexy, fine. But the underlying foundation is the base is your
right.
basics. Get
Yeah.
your 6 to 8 hours and you'll be fine.
Yeah.
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 I
I
mean the
think
same is true even regarding like consistent and safe exercise, right? So like or
yes.
even consuming a nutrient diverse diet. So like I said, each individual behavior has ridiculously complex underpinnings associated with it. They don't need to be complicated by jargon that we're not effectively explaining to people, especially on social media, which is why I put a lot of time into this.
Yes.
Um, but good sleep, exercise, good diet for you.
Yeah.
Don't be an alcoholic.
Yep.
Don't inhale combustible material.
Yeah.
Gets
I'm
me the
hearing all the
58,000
basics.
followers, right?
Yeah.
It's
Yeah.
it's the stuff everybody knows, but just doesn't want to do.
Yeah, and I think this
Yeah.
rebranding on social media where, they use fancier terms or more elaborate words, and it boils down to the same stuff, that, having cancer prevention is going to be about having a healthy lifestyle, putting good things in your body, you
Yeah.
know, eating your fruits and vegetables, exercising, all the boring stuff that have been there for decades,
Yeah.
right?
The,
And
the thing is though
there's
that I'm trying to point out here, and I'm sorry to cut you off.
no, you're good.
We say it's boring, and it is
Yeah.
conceptually, but like when you actually think about these
What?
things, they're not boring at all.
Yeah.
Like if we talk about sleep health, the things that enable us to understand sleep health are all the cool scientific mechanistic shit about circadian metabolism. So if
Yeah.
people care about learning sleep health and why it's 6 to 8 hours, they can dig deeper. Why is the suggestion 6 to 8 hours? Why are diabetes, obesity, and many cancers now associated with sleep dysregulation? They can find those papers there, but
Yeah.
it's not just this superficial statement, right? We have to think deeper about the things that actually enable
Yes.
good health. So while it might be boring to say consume a nutritionally diverse diet, we have to dig deep into what that means for an individual person, right? So like I won't have the same diet as somebody who maybe has a different work-life balance. Maybe they have other health predispositions. Maybe they have phenylketonuria, a
Yeah.
disease where you can't consume phenylalanine
Yep.
Yeah.
highly and abundant in food. Maybe they have some sort of rare genetic disease. Maybe they have Lynch syndrome where they need to be more conscientious about the fiber that they consume. Like
Yeah.
there's context here that while the basics sound boring, there's enough complexity there to go around. We don't need to
Need to
rope
add to
people in
it.
and manipulate them into feeling smart without understanding it. And that's why I do what I do. And
Yeah.
like I said, it's gotten me 58,000 followers, of which I'm super thankful for. But
It's
like
a lot, man.
it's much more than I was expecting doing what I do. But the thing is, I know at the end of the day that I could have a million by now if I
Yeah.
just sought clout,
Yeah.
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 right? And maybe sacrificed a little bit of my moral and ethical compass.
Yeah. Yeah.
I was just
Um,
gonna say I feel the same way often, like especially with Vinny's line of stuff. I'm like, man, if you just decide to put out a supplement,
yeah,
Not
dude,
doing it. Not doing it.
it's depressing.
I'm
it's
not doing it. I'm not.
but
Well,
I'm
if
Not
you
like
going to
ever
the dark
do
side.
it,
I'm not
if you
going to the
ever do
dark
it,
side.
man, I swear I'll find you. I'll slap you. We don't know each other, but I
it's
will
gonna be
find you and
called
slap you.
creatinine.
Yeah.
Yeah.
Oh man. But I will say though, like the best thing you said in this last paragraph of you speaking was that biohacking is shit, right? Because that's like that's the buzz term that gets thrown around and everyone hears like because now peptides and biohacking, right? So
Yeah.
and it's funny because like I put out a post, I think maybe like 2 weeks ago, I was like, biohacking doesn't exist. Anybody who says that they're a biohacker, that's a red flag. It's bullshit. Don't listen to them. And then you get these so-called biohackers that are like against vaccines. But if you
Yeah.
really want to look at what biohacking might be, it would be taking a vaccine where you can
I
improve
think
your immune
Vaccines are the
that's
system.
best way to biohack.
You
It's
can you can
literally
improve your
made.
immune system by giving yourself a little bit of a dead virus and it builds a better immune response to it. That would be considered biohacking, would it not? Like
No, that's exactly. You make a post on that. Vinny, I swear to God, let's collaborate on it. We'll do it.
okay, we'll do that. We'll do
That's
that
We'll
one.
make a
Yeah.
post. But
post,
no, that's
guys.
literally it. Like a vaccine is no better way to biohack.
I
Yeah.
mean, literally, and they're against it. And it's like, wait a minute. So I have to worry about,
Contradictory.
yeah, I
It's
have to worry
because they
about
don't
mental illness.
understand what they're talking about. That's the whole
Yes.
point. If you
Yeah.
Yes.
understood what you were saying, you would know that what you're saying makes no fucking sense.
Yes.
Yes,
Yeah.
exactly. Yeah.
Ah, I'm just
So
glad someone
biohack
else said it.
this.
Yeah, yeah. That's gonna be the cover
Yeah,
of
just
this episode.
there's a snapshot for you.
Oh man. Well, look, Joe, thank you so much for your time, man. This was great. We went a lot longer than I expected, but it didn't even feel that long. So this was a
This
really
was one
great
of the
conversation.
funnest, yeah, this is one of the funnest podcasts I've done,
Good.
if not the
We're gonna
funnest.
do another one, man.
Good.
Just in the future, we're gonna do
Man,
another
I'll
Yeah,
one.
talk to
like
you
Like
guys
that.
anytime.
Yeah.
It's like,
we need to sit down. I want to hear more about like your book. Essentially, we're going to do another one where we're just going to talk about your personal story because I feel
okay,
like you're such a unique person, and I want to know more about your story. I
Yeah.
really do. Like this is so interesting to me. Um, your depth
okay.
and complexity in your own life and how you bring that forward and the work that you do, I think is so great. So please continue to do the work that you are doing.
We'll have some wine and beer. We'll drink while we're talking about it.
Okay,
So
what
keep
kind of
some
wine
people.
though?
I like Pinot Noir.
Okay, I'm a Chianti guy,
Oh,
so
Oh,
okay. Yeah,
nice.
yeah. So we
okay.
stick with the red family.
All
Yeah.
Yep.
right. How about you,
Yeah.
Aaron?
You know what? I just started drinking not too long ago, so he got me into the red wine. I'm barely like starting to digress. I didn't drink until my mid 30s.
Yeah.
So,
Okay, good. I mean, this is a cancer-focused podcast, so I will say for anyone, drinking is not good for you. As I
No,
had
yes,
it's
mentioned
not.
earlier, alcohol consumption is not good for you, but you can handle it in small quantities. So
yes.
Yeah.
And for some
keep
people
that in
on the
mind.
spectrum, I feel like it might be a little therapeutic.
Yeah, with your social aspect of it. Yeah, I
Yes.
could see that.
Yeah,
So Joe, why don't
absolutely.
you tell everybody where they could find you, where they could catch up on all of your posts, and if you have like a site that puts out your latest research and stuff and whatever you're working on, lay it all
Sure.
out for them so they could go follow you.
Sure, yeah. So people can follow me on Instagram is where I do almost all of my posting, and that's at Dr. dot Joe Zundell, and we can tag it in the show notes or whatever.
Yep.
Um, and I also have a newsletter. There's links on my page in my link tree. So people can click that. They can sign up to my newsletter. I am affiliated with some supplement brands, but only because I trust them 100%. And I don't want to feed people bullshit if you didn't gather any of that from this
Awesome.
episode.
Yeah.
Um, so if people want to, again, I don't force anything on anybody, but if people want to look at those things, they can. But ask me first. I won't suggest anything I wouldn't do myself to people.
Yep.
Again, integrity is super important to me, above all else. And I have a website. It's drdrjoezundell.com. They can go there. They can check my CV. I should probably update that a little bit, but it's there. Um, and just message me if people have questions. I'm pretty responsive on Instagram. Um,
I can attest to that. Very responsive.
yeah. Again, everything that I do is about comprehension. So I think a lot of people place too much emphasis on this set it and forget it approach to Instagram,
Yeah.
where they just post. But the most important thing people can do if they want to educate people is in the comment section.
Yep.
Yeah.
Because how bad of a teacher would I be if I just went, like, think about it. You go into a classroom, right? And you present PowerPoint slides and you allow no questions. Like, do
True.
you expect your audience to understand what you're doing? No, you have to allow them to interact. That's why it's called social media, not whatever the fuck would be the opposite of that.
Unsocial media.
Yeah.
Anti-social media. So
That's
yeah, so
interesting. I never really thought about that.
Yeah,
That's a
yeah.
good way to put it. I
And
love
it
a
that.
lot
pushes
of
your
science
post on the
communicators have.
algorithm. It
Yeah.
pushes your post through the algorithm, the more comments and interactions you have. So,
Does it?
yeah. I don't know.
Oh, it's
Hey,
gotten really
you
bad
were like,
over
hey, how did
time.
I get to 58? That's how.
That's not recent. I will say, like, whatever's going on Instagram the past year has been abysmal for me.
Me too.
Yeah.
I
Dude,
don't get any interactions on my posts. And I'm like,
yeah, I
I
used
thought
to
I put
like
out good content.
I put so like yeah, you put out amazing content, but like you should see some of my older posts. They sucked
Yeah, yeah,
in comparison to what I do now. And that's unbiased. Like I say this wholeheartedly. They sucked.
yeah.
And you look at the interactions on those, and I had maybe 1,000 followers, maybe two back then. And some of those posts have like 400, 500
Yeah.
likes on it, 20,
Wow.
30 shares. Now I have 58,000 followers. Nobody fucking sees when I post.
Yeah.
And I'd be lucky to get 350, 400 likes unless I'm super dickhead about something
Yeah,
and
yeah,
get
yeah.
people to argue aimlessly about shit, which I
Yeah.
refuse to do anymore.
Yeah,
Yeah,
yeah.
yeah,
I
yeah.
went
So
down that route and it didn't resonate with me. It was just
it
a bad
feels
feeling.
bad.
Yeah,
Yeah,
yeah.
it's a bad feeling. And I just was like, I can't do this that much anymore. My friends loved the content. They were like, yo, you
Yeah,
got to keep calling these people out. I'm like, I
that's what
just don't,
everyone tells me
it
too.
don't feel right. Yeah.
Yeah.
It just
It's
doesn't
It's
funny.
feel
He
it's
right.
wants
also
me
like
to do that,
hard
and I'm
I
to
like,
want her
no.
to do it.
Yeah.
Because she'll say it to me and I'm like, do it to them.
I'm like,
Oh my
I
God.
think I think there's tastefulness in doing it occasionally.
Yeah.
yeah.
But if it's like your whole image, there are some of these science communicators where their whole page is like, oh, they just call out anybody,
Yeah.
whether it's a
Yeah.
scientist, they're scientists themselves, right? Or in
Right.
the health profession, they just fucking call people out. It's like if that's your whole image, you can't tell me that you give a shit
Yeah.
Yeah.
about like actually teaching people. You
Right.
can't tell me you do.
Yeah. Yeah.
Because and also it's like I care so much about what I do and I attach maybe more than I should of my own feelings to stuff. Um, but like I have to enjoy what I do.
Yeah.
Yeah.
And if I'm making content that just seems like it's a slog to make just because it's what people want or what they think they want, which is just me yelling at a camera, which is essentially what I've done this entire time on this podcast, so excuse me.
In
But
a good way.
if that's what people want, I'm sorry. I've got blood pressure issues as it is. Like
Yeah.
Right.
I don't like this is a hobby to me. Nobody's paying me to give you my education to you for free. I'm gonna try and enjoy myself. And if occasionally I swear or call someone out, and so be it, enjoy it. But like let's all get used to sharing the impactful stuff, the stuff that we put actual time and effort into that actually educates people instead of, oh, let's call out the glucose goddess for the fucking 5,000th time,
Yeah,
expecting
no.
that her and her audience will change their views all of a sudden, or Paul Saladino or any fucking Gary Brecka. Like,
Yeah.
what makes you think that their audience isn't a cult? You think you calling them an asshole is just gonna match to be like, oh, they are an
That's
asshole.
what I say. That's what I say. I'm like, that's not gonna change anybody's belief. Like they believe that
Yeah.
deeply.
And then they want us to make these videos calling these people out. It's like you don't understand how this works.
Yeah.
Yeah.
You walk into a cult following and just be like, guys, stop. And
Yeah.
they do it.
Yeah, exactly. That's exactly how I feel.
But Joe, thank you so much once again. Really appreciate it. We will have a session 2, and I will make sure that everything is in the show notes for everyone to go follow you, man.
Yeah, happy to hang out with you guys more. This was super fun again. And thanks again for having me. And like I said, if your audience needs anything, I answer questions. So please ask me. There's a lot that we cover here today that still I don't understand to 100% certainty.
Yeah.
Yeah.
And
Same here.
with back and forth, we can try and understand it all a little bit better.
Yeah, I love that.
Appreciate that.
Yeah,
That's a
thank
good
you.
that's a good quote to leave on.
Yes.
Yeah.
So on behalf of Balanced Bodies, we just want to say thank you for joining us on this episode of the Balanced Bodies Blueprint.
We are committed to bringing valuable content. And if you enjoyed today's episode, we'd greatly appreciate it if you can take a moment and like it and leave a 5-star review.
On Apple, just go to the show, scroll down to the bottom and rate it there. If you're on Spotify, go to the show's page, click the 3 dots, and you can rate it there as well.
And if you believe in the power of knowledge, share this episode on your social media to try and get the information out there to as many people as possible.
And as you navigate your own path towards better health, remember that Balanced Bodies is forever in your corner. See you all next week.
The podcast content may include discussions of medical topics and health-related information. However, the information provided should not be considered exhaustive or complete, and it should not be relied upon as a substitute for professional medical advice or treatment. Always seek the advice of your physician or other qualified healthcare providers with any questions you may have regarding a medical condition or treatment.